Last year I talked about adult glioblastoma, and I wanted to kind of bring it to something a little bit different that I'm particularly focused on right now, which is pediatric glioma and brainstem glioma in particular. And I've titled this Therapeutic Strategies for an Incurable Cancer, and that's exactly what it is, unfortunately. One of the first patients I ever saw at UCSF in 1985 when I came there was a child with brainstem glioma from Indonesia, and I have to say that even now, after all these years, the outcome's really not changed. It's a very difficult problem. So I'm going to go very quickly through this. It's more clinical than translational, but and I'm going to talk about the kind of the strategy we have through what's called the Pacific Pediatric Neuro-Oncology Consortium, which we started about 4 years ago with a couple of like-minded pediatric neuro-oncologists who felt like we had to do something different to try and make a change. So high risk, high reward, do something we hope is significant, and take chances to try and make a difference in the disease. And we decided to do this outside of the context of the NCI because we felt we would have more freedom to do that. And that has challenges of its own. So this was our strategy about 3 years ago in terms of how to tackle the disease, and I'll tell you why we got to that in a minute. But basically, we wanted to have a precision approach. We wanted to have a convection-enhanced delivery approach because of the issues about drug delivery, and we wanted to have various immunologic approaches. And we set out to try and do that, not only to create the trials, but also to get the funding for it. So for those of you who don't deal with kids with brain tumors, they have a wide spectrum of different kinds of diseases, and a lot of them are very well managed. A lot of them are what we call lower grade tumors, and the idea here is to take care of them without hurting them, but there are about a half of them that are gliomas, and a small portion of those gliomas are high grade. And the brainstem tumors represent about 12 or 13% of those gliomas. But if you look at the cause of death, that small group of children represent a disproportionate number of children who die of brain tumors. So that's the disease, and location. I'll have a slide about that, but the disease originates in the pons. You can't live without the pons. And it's very clustered among children who are between the ages of 5 and 10. I mean, you rarely see it outside of that age group. And it's a difficult process when a child comes in with their parents. They have a very short clinical history, scans done, DIPG, diffuse intrinsic pontine glioma. The parents get on the web and they know immediately that their child is going to die. And that's essentially the decision-making process when you talk about different options. What can I do so that my child will live? Median survival has not changed in 50 years. And the median survival is about 9 months. When a patient progresses, which is usually in 6 or 7 months, they usually only live about 2 or 3 months after that. So that's worse than glioblastoma. The disease, fortunately, is not common, only about 300 or 400 kids per year. So it's a very rare disease, but it's obviously very important, right, for every child that comes. In the past, what we've done, which is very empiric, we would take drugs that were used for adults and then put them in kids and assume that those gliomas in adults would translate into therapeutic success in children. And that's always been wrong. This was a quote that Nalan Gupta, one of our pediatric surgeons, found in a chapter in a book back in 1996. That was the story back then. And because we thought we were so smart with MR imaging, the decisions through the Children Oncology Group and other groups was use the MRI scan, don't get tissue. So for the last 40 or 50 years, people have not been getting tissue. So we never knew the biology of the disease that we were treating. We were just treating an image, an MRI scan, and never figured out what the underlying biology was. Back in 2006, as part of the Pediatric Brain Tumor Consortium, we felt that we had to change that paradigm and to get tissue from all of these kids because we didn't know what we were treating, the underlying biology or the disease. And at that time, CTEP, the NCI said no, it was too risky to do that. When in fact, children had been getting biopsies of their pons for many years before that. And in Europe, they were way ahead of the curve getting tissue, and they were starting to understand the underlying biology. But we couldn't get it through. Finally, we opened up a philanthropically supported study that came out of Dana-Farber. But the point here was it took 6 to 10 years to try and even start the process of getting tissue to help us understand what the disease was we were doing. And the trial that was attached to that early study was just basically using information from adult glioblastoma and hoping that it would make a difference. This trial was just completed last year. We have 25 institutions that were involved. What came out of it more than anything else was the genomic profiling that we were starting to understand. And it turns out that there is a significant difference from glioblastoma. Even though these children ultimately die of what's called glioblastoma, it's very different than glioblastoma in adults. The genomic underpinnings are completely different than what you would see in adult glioblastoma. And all of the therapies that we were using in the past that were focused on adult glioblastoma, of course, wouldn't work. Of course wouldn't work. The other issue about pontine gliomas is that in some brain tumors, in effect, these are kind of bland in terms of genomic alterations. So we say that there's heterogeneity, there is, but the kinds of mutational loads or the alterations that occur in these children are actually quite small. And it's interesting that that these children die so quickly with a disease that isn't so genomically complicated. But that's truly the case. About 4 or 5 years ago, a critical piece of information was finally uncovered based on the fact that children were now getting tissue, and the underpinnings that genomic profiling, good platforms for looking at alterations were coming into play, and an epigenetic mark was discovered called the H3.3 K27M mutation, which is really only seen, ultimately in children who have brain tumors and brain tumors that are in the midline. So this is an epigenetic regulator that was discovered just very recently, and an awful lot of emphasis is now being placed on what to do with it. This mutation that's found in DIPG changes the amount of methylation, if you want to think, the trimethylation that occurs near the epigenetic regulators, the PRC complex, and it changes globally a gene expression both up and down along the genome in a very, very complicated fashion. But this is actually an important target. It's one of the few mutations that occur in DIPG. It occurs in about 70 or 80% of children with DIPG. When it occurs, those children have the worst prognosis. They do worse than children who don't have that mutation that still have DIPG. So it's an important finding. It's one of the very few critical findings that have actually driven the way that we think about the disease. And you can see in this kind of spectrum here that it's restricted to children, it's restricted to diseases in the midline, and it has the worst prognosis of all the gliomas that you can see here, adult as well as children. And topographically, this is where these diseases occur in the midline. So we actually now are starting to think about not only DIPG as being restricted to the pons, but a spectrum of diseases in the midline thalamus, pons, that all have the same unique biologic characteristic and driver mutation called the H3.3 K27M mutation. So knowing that, what do you do? Well, the ideal pipeline would be that now that you understand a little bit about the biology, you can start to create model systems and then do clinical trials. But we wanted to do this as quickly as we could to make things happen. This was a child that we saw at UCSF about 7 or 8 years ago with DIPG. This little girl underwent a series of biopsies. We developed a good xenograph. The xenograph had the K27M mutation, and we were able to propagate the xenograph and not only have it available to use in the flank or in culture systems, but we could actually place the tumor in the ponds of rodents so that we had orthotopically relevant tumor model that we could start to follow, and it was modified so that we could use bioluminescence. And this cell line and this cell source has been shared around the country, and now since that's been published, there are about 10 or more, and we're developing new cell lines and xenographs on a regular basis now. So I want to get down to precision medicine. I talked about that last year in adults, but even though there are a few alterations and mutations in this disease, there is heterogeneity. Every child is a little bit different. And again, we can't use a one-size-fits-all approach like Dr. Hood was talking about this morning. So we wanted to develop a precision approach for these children, and we modeled it over the Ivy trial that I did in adults, which was to genomically profile and take that information and make a decision, and then try and treat these children with more than just one drug, both positioned and repositioned agents. Again, taking from Dr. Hood's comment that one drug is probably not going to make a difference, and so we allowed the option of doing many studies and open this many drugs and open this protocol about 2 years ago now, called PNOC 003. And the workflow and the way that we did this was essentially the same as the adult study. Is when a child came in with an image that looked like DIPG, those children would get biopsied before any therapy, and typically we would do 4 or 5 biopsies in that child at the same setting. And then we would send the tissue fresh for profiling for whole exome and RNA-seq, not whole genome, but we're doing the whole genome now. And then we would match that to alterations and drugs that could fit the alterations and pick 3 to 4 drugs that made a sense. And again, positioned or repositioned agents, up to 4. And that was our feasibility study. This is the workflow that went through that. We could get the information within 3 to 4 weeks. We would convene a molecular tumor board with every child that was biopsied. And then we had a neuropharmacologist and a number of clinicians and genomic scientists that would help us organize that. We would create a report. It would be part of the medical record. And then we would treat the patient after radiation therapy. We also made the decision to create xenographs of all these kids, and we get circulating tumor DNAs or liquid biopsies all along the way so that we could try and develop biomarkers as well. So we went through that process. We had a feasibility study. The first 15 patients, this is the profiling that came up, and you can see again that there is heterogeneity in the alterations that are there. The H3.3 more common than any other alterations, but also alterations in P53 and other things. Some things that aren't really druggable. And oftentimes we're making decisions not only on the mutational abnormality, but also the pathways that were altered in the hopes that we could integrate both somatic mutations with pathway alterations and then treat the children. And these are some of the drugs that were used. So that trial ended about 4 or 5 months ago, and then we amended the protocol to make it an efficacy study so that we now, instead of giving the option for treatment, children are being treated with the therapy that we recommend, and we're gonna look at outcomes. And again, this is a rare patient population, only 300 or 400 patients per year in the United States, so we can't do controlled clinical trials. So we're going to do the best that we can with targets in terms of efficacy and see if we have a hit. And then we're going to go to a master protocol design and hopefully try and get investigational agents added into it. So that's our precision approach. So after that, what do you do? So again, the issue for us is drug delivery. How do you get drugs into the brain, and how do you document that you get drugs in the brain? The blood-brain barrier is very adept at keeping things out. Less than 1% of almost every drug ever gets into the brain, and you never know whether they hit the target and it modulates the target, and what happens after you perturb the target. So how do you deal with that? So we started about 10 or 15 years ago trying to focus on drug delivery issues, both convection delivery and intranasal delivery, as well as systemic delivery, and then trying to formulate ways of trying to enrich how much drug gets in and how long it stays there. And we've been working with nanoparticles for that reason and model systems. We actually have been doing trials with a construct called a nanoliposomal CPT11, so irinotecan, an old cytotoxic agent, a topo inhibitor, and have done some phase 1 systemic studies. But we wanted to say, okay, could we use that strategy to get that drug into the brainstem of children, and why would we want to do that? Well, we know topo-1 expression is overexpressed in adult glioblastoma from the TCGA. From the trial that I just described, all those kids had tissue, so we interrogated the tissue to look at topo expression in those kids' tumor, and it's very highly overexpressed. So we know that the target of irinotecan, the topo, is there. And we've done studies now in model systems looking at the delivery not only of CPT11 into the brain, but CPT11 encapsulated in the nanoparticle, and it's better when you do it that way. And if you do it by convection delivery, it's even better, so that you can optimize drug delivery to a target that you know is actually there in a kid with a brainstem tumor by giving it directly. And we use that model system and we use that child's xenograph to do this kind of work. And there's a dose response, and it looks like it's a reasonable thing to do. And we've compared intranasal delivery, systemic delivery, and intratumoral delivery. And intratumoral delivery is clearly better, so why do anything else? So the issue again is, can you put catheters in the brainstem of children and give them a drug? Well, yeah, people have been doing that now recently with a number of different drugs. We decided that doing it once was not enough. And so we set out to develop a protocol where, knowing that the residence time of this nanoparticle is measured in weeks, if you put CPT11 in the brainstem in a model, a rodent model, it stays there for like 6 weeks and slowly is releasing that irinotecan. But then it's gone, right? The concentration goes down. So we felt like in order to make a difference in children, we should do this every 4 to 6 weeks. So we went to the FDA and asked them to let us do that, and they said, well, do you think it's safe? So yeah, we think it's safe. So we proved that it was safe, at least in dogs who have tumors, and we have a good connection with the vet school in Sacramento. And these dogs get a lot of brain tumors, and we've looked at convection delivery of this nanoparticle in dogs. We've been able to optimize the technique and the tools and the catheter systems and the placement of the systems with real-time imaging using gadolinium to let us know that the delivery is going to be where it is without any backflow. And we've seen regression of these tumors, these spontaneous dog tumors, and we've done it over time. So we had the data to support taking that high risk for a potential high reward in children, and went to the FDA to do that. It took us 2 years in debating this with the FDA, but they finally gave us approval to do that. We also, so the idea is brainstem glioma. We know that the target is there. We'll get tissue from those kids. We'll confirm that the target is there. They'll finish their radiation when the tumor volume is small, and then we'll put catheters in at the end of radiation and start with real-time imaging, deliver this nanoparticle of CPT11 into those tumors, and then every 4 to 6 weeks continue it until they progress. So this is just a platform, and the platform is important because if you can do this and you can show the FDA that it's safe and you can show to families that it's reasonable, and if it makes a difference, that would even be way better. But that could be a platform for putting multiple agents into these nanoparticles or multiple drugs and leave permanent catheters into these kids and convect them over time so that you could have 3 drug cocktails or 4 drug cocktails or other agents that you can put in that. So this is the beginning. We're very excited about doing this, but it's technically challenging, and every kid that we do, it costs a ton of money. So and we're philanthropically based, right? So there's no drug companies that's going to pay for this. So we did get a foundation to give us $2 million to do this in the first 7 or 8 kids, and then hopefully 10 kids. And then maybe if we show that the platform works and it's reasonable, we can get additional support from maybe a drug company. It's hard to say. I mean, there's no profit margin in taking care of a kid with a brainstem tumor. There's just none. So you can't get companies to do that. But we did have a company who gave us the drug, and that's great. And we did get a philanthropic partner, and that's great. And we're going to start this process, and we hope it'll make a difference. And then finally, this issue about immunotherapy and targets. The K27, the H3.3 K27M mutation actually turns out to be a neoantigen, and it is recognized on the cell surface, and it allows the development of novel peptide-based vaccines, and it's homogeneously expressed in tumors if you just look at tumor tissue from these children. So it's probably a founder mutation, it's probably a driver mutation, and it's probably critical for the development of this tumor and the propagation of this tumor. And Hideo Hokata in our group has worked on very specific epitope binding sites in an HLA-restricted fashion context, and has actually shown preclinically that he can make a peptide-based vaccine that would target specifically these tumor cells. And we finally, again, got approval from the FDA to do this. This is interesting in that it's a first-in-human study. It's a first-in-human study being done in children with brainstem tumors. And so the FDA was a little nervous about it. We're a little nervous about it because you always worry about allergic encephalitis or other problems when you give vaccines directed towards a target in the brain, and then hope that there's not going to be some problem with damage to the nervous system of these children. But we did get this approved. We also got it funded through philanthropy to do this, and we've got a company to create the vaccine. We have the IND in place. This took us about 2 years to get through the process of approval, and we got approval last month. And and as soon as we released that the trial was open, we had children and families, contacting us. And that's one of the problems, because we could only treat 6 patients and then wait. The FDA said you can treat 6 patients, but then you have to wait 3 months. So we've done this 6 patients, and we have to wait 3 months to make sure that it's reasonably safe. But it's very exciting because we have this founder mutation, and it's expressed in these children, and it's one of the few alterations that you have. And using a vaccine hopefully will be non-toxic, and we'll target exactly that epitope, and hopefully it'll make a difference. So, and then the final thing I'm going to say is, because I know my time is up, is we also are interested in this target because there actually are drugs, Jumanji D3 demethylase inhibitors, that exist. This is actually, this alteration in epigenetic in the epigenome is actually common in a lot of different cancers, not particularly this mutation, but but this is druggable. And we're working with Pfizer now because of preclinical research that Rintaro Hazijumi, I have a polymorphism, I can't say his name, did with that child's tumor, showing knockdown with this demethylase inhibitor with decreased proliferation and apoptosis that was restricted only into the tumors that had the K27M mutation. It's highly specific and highly effective, not effective at all in the non-K27 wild type group. So we're now working with Pfizer to develop a brand new drug that is CNS penetrable, to actually get their chemists to make this drug in a platform that truly gets into the brain. And we've been working with them for about a year and a half now. They're very keen on helping us to develop a drug that we can use, and they put their whole medicinal chemistry group on it. The plug for them is that they're doing it, but they also hope that it'll work for glioblastoma and other cancers as well. But we'll probably have a first-in-human trial in pediatrics with the K27M mutation. And if it doesn't work too well, we're also working on a soluble form that we can give by convection delivery as well. So it's really exciting. And I only put this up to say everybody's doing PD-1 inhibitors, so we are too. And we have a company now that's working with us called Regeneron with their PD-1 inhibitor that we're just getting ready to open, where kids with DIPG will get their PD-1. But all those kids are going to get a biopsy before we treat them. So we'll at least know something about their tumor and whatever biomarker may correlate with outcomes. Again, we don't have randomized trials in DIPG. There are just not enough patients. And I don't think parents would be very accepting of a randomized or a placebo-controlled study, but we are doing that. So we have a spectrum of approaches based on high risk, high reward, I hope, that are significant based on tissue correlation, biology, and maybe it'll make a difference. And I started out with a strategy through the PNOC, and Dwayne will know this very well. We also had a dendritic cell vaccine trial that we wanted to do, and worked with the company for a long time before they pulled out. That's a problem. It's a problem to get engagement of pharmaceutical groups for diseases like this, because they're so rare, and they're worried that children will get hurt, but they're all dying. They all are dying. So you've got to take high risk and high reward. And anyway, so I'm very pleased with this portfolio of protocols that we're doing. The last slide is this. The NCI. This is, it took me a while to get all this data, but the NCI does not support a lot of pediatric cancer research in terms of their whole portfolio. And when I started to filter down from what the NCI gets into what actually gets into pediatric cancer of all of their funding, it was only about 4% of the portfolio is in all pediatric cancer, not just CNS, but pediatric cancer. Less than 1% of the budget goes to CNS tumors, less than 1%. But the number of years of life lost of a child is huge. It's huge. If a child dies at 9, then you've lost 80 years of life. And every child that dies, that's another 80 years. I mean, when you start thinking of years of life lost, it's huge. And other gliomas in children as well. So it would be really nice if there was more funding and that's why we deal with philanthropy, because that's where the source of revenue is, to do the research that we're trying to do, and these are expensive studies, and occasionally some pharmaceutical groups, but not too many. But anyway, we're hoping to make a difference. We're hoping to be a bit, significant as we can in this very, very rare incurable disease, to see if we can push the mark, and hopefully in the next couple of years we'll have some data about this that'll, I hope, make a difference. So thank you.